Welcome back to the Deep Dive. Today we are diving deep into the human genome, and we're tackling what you could call a modern paradox. I mean, sequencing a genome is now incredibly fast, it's cheap, but interpreting it, that's still incredibly slow and really difficult. We've sequenced millions of people, but we're just drowning in data. We're
Yeah.
talking about these things scientists call variants of uncertain significance, or VUS. We know these little variants exist in our DNA, but we have no idea if they're just harmless background noise or if they're the root cause of some life-altering condition. And traditionally, the computational methods have tried to give us a really simple binary answer. Is this variant pathogenic or is it benign?
And that binary view is it's completely inadequate for the true complexity of human disease. It really is.
Oh, so.
Well, when you treat all pathogenic variants as one big sort of homogeneous class, you completely miss the forest for the trees. I mean, think about it. A mutation that causes a minor correctable eye abnormality gets the same label as a mutation that causes a lethal, severe developmental disorder in a child.
Right. They're both just pathogenic.
Exactly. And the current tools, they are fundamentally incapable of giving us that high-resolution picture we need, especially when you're dealing with variants outside of protein coding regions or just trying to judge the difference between a subtle disruption and a catastrophic functional collapse.
Okay, so let's unpack that. Our mission for this deep dive is to explore two what I think are revolutionary machine learning models that specifically tackle this interpretation challenge. We're going to be looking at how V2P and popEVE are moving past that simple binary prediction to really accelerate genetic diagnoses, particularly for rare diseases. They do this by mapping variants to specific disease categories and, crucially, predicting their severity across every single protein in the human body. So let's start with specificity, understanding what a variant might actually cause. And that brings us to the first model, Variant-to-Phenotype, or V2P. This is a multitask, multi-output model. It can look at nearly every kind of variant, from single letter changes, SNVs, to insertions and deletions, and it does this across the whole genome. But its big breakthrough isn't just saying a variant is bad, it's linking it directly to top-level disease outcomes.
Yes, and what's so fascinating here is how V2P achieves this. It's all about conditioning its training. So instead of just training it on pathogenic versus not pathogenic, they force the model to predict which type of disease it causes. The output is really extensive. You get a general pathogenicity score, plus you get likelihood scores for 23 top-level disease categories. And these come from the Human Phenotype Ontology.
Okay, hang on. Let's slow down on that term for a second. Human Phenotype Ontology, or HPO. For our listeners, what exactly is an HPO, and why is organizing variants by HPO categories such a revolutionary step?
That's a great point. The HPO is essentially a standardized vocabulary. You can think of it as a common language that clinicians use to describe clinical abnormalities.
So it's a structured list of symptoms.
A comprehensive, structured list of symptoms and observable traits, exactly. So by teaching the model to predict which of these 23 top-level categories, we're talking big buckets like abnormalities of the nervous system or neoplasms, a variant is likely to fall into. You're providing the model with essential biological context. You're forcing it to learn that the genetic consequences leading to an eye disorder look really different from those leading to a heart disorder.
I see.
And this conditioning, this multitask learning, it dramatically improves the quality of the overall prediction. Because it's not learning one big, vague class of disease, it's learning 23 distinct mechanisms of harm.
And to make these predictions, V2P is taking what you could call a kitchen sink approach to its features, right?
Absolutely. It's a true kitchen sink. They're feeding it every piece of biological context you can imagine.
Gene function data, detailed protein structure predictions.
Evolutionary conservation across species, tissue-specific gene expression, epigenetics, even phenotypes they've observed in homologous mouse genes. It's an enormous data feast.
And analyzing all those features must reveal some really interesting patterns.
It does. It reveals some truly foundational biological patterns. We often talk about genes being essential, and V2P provided some really concrete evidence of how that relates to the type of disease. For instance, V2P found that variants causing really severe centralized problems like neoplasm, so tumors or abnormalities of the cardiovascular and nervous systems, they show a significantly greater tendency to inhabit what we call essential genes. These are genes that are highly intolerant to a loss of function. If you mess them up, the whole system can collapse.
So if I'm getting this right, these are kind of like the core internet routers of the cell. If one of them goes down, the whole system failed.
That is a perfect analogy. They are critical central pathways. Now, contrast that with variants linked to abnormalities in more peripheral systems, phenotypes of the ear, the eye, the genitourinary system. V2P found these variants are statistically much less likely to inhabit those highly essential genes. And this just makes perfect biological sense. Messing up that core router is devastating, whereas, impacting a single user's mouse or monitor a peripheral system, that results in a very specific, contained problem.
That's a brilliant way to put it. And I assume the performance of the model really bears out this structural distinction.
It absolutely does. When they were testing missense variants, V2P's phenotype-specific predictions, they actually surpassed its own general pathogenicity prediction score.
Wow.
And it surpassed all the other compared methods for 21 of the 22 phenotypes they tested. So it's a huge validation of the core idea. Separating pathogenic variants into specific categories fundamentally improves your predictive accuracy.
Okay, so VDP helps us define what a variant might cause, but that still leaves a pretty big gap, doesn't it? I mean, if VDP flags two variants in two different genes as highly likely to cause, say, a neurological abnormality, how do you compare them? How do you know if one causes mild tremors and the other causes lethal infant encephalopathy? That's where this idea of calibration and severity comes in, and that brings us to popEVE.
That is the crux of the problem that popEVE solves. It's about comparing variants across entirely different genes, the challenge of proteome-wide calibration. A score of, say, 0.9 in gene X might be terrible, but a 0.9 in gene Y might be survivable. So popEVE's mission is to provide a continuous, calibrated measure of deleteriousness that is comparable across the entire human proteome.
And its architecture sounds really unique because it's a hybrid approach. It's marrying sort of the ancient with the modern.
Precisely. It starts with the ancient. It uses deep evolutionary data, leveraging scores from models like EVE, which have been trained on protein sequences across billions of years of life. And this establishes a basic, highly reliable fitness score. How conserved is this amino acid, and how likely is a mutation here to break the fundamental function of the protein globally?
But that's not the whole story.
No. Then it introduces the modern human-specific element. It combines those deep evolutionary scores with shallow human population data from sources like the UK Biobank and NomeAD. This is done using a technique called a Gaussian process. You can think of this Gaussian process as a kind of sophisticated calibration layer, a tuning dial.
Okay.
It takes that massive universal evolutionary score and it adjusts it based on what is actually tolerated or constrained, specifically within the modern human population. This makes sure the score reflects not just general protein function, but human-specific constraint and severity.
So it's basically asking, is this mutation tolerated in bacteria and in the UK Biobank? And if the answer is no on both counts, the score is extremely high.
That's the idea. And if it's tolerated in bacteria but very rare in the UK Biobank, the human data will pull that severity score higher.
Which leads us directly to the critical test of severity.
Yep.
Did this calibration layer actually pay off in terms of clinical impact?
It did, and spectacularly so. The sources show that popEVE scores significantly separated pathogenic variants associated with childhood death from those associated with adult death. And it did this better than every single other tested method.
That separation is a huge piece of evidence, right?
It's crucial evidence. It proves that popEVE isn't just identifying protein disruption, it is capturing the severity of that disruption on the level of the whole organism, right down to life expectancy.
And this capability, I assume, leads to a massive reduction in false positives, which is, arguably the biggest headache for clinicians who are interpreting genomes.
Absolutely. Think about the noise reduction. If you set a high-confidence severity threshold using popEVE's metric, you can successfully recover 50% of diagnosed severe developmental disorder cases, which is excellent. But at the same time, you're predicting that only 11% of healthy UK Biobank individuals carry an equally severe variant.
That's powerful. You're filtering out so much noise so effectively.
Correct. And you can contrast that with other state-of-the-art prediction methods. If you run that same test using them, they might classify 44% or more of the healthy population as carrying variants of equivalent severity.
Wow, 44%.
Think about the clinical utility difference there. The older models were, raising the alarm 5 times a day for a healthy person, maybe averaging 5 pathogenic hits per healthy genome. popEVE is raising it less than once a year.
That's the difference between being clinically useful and just being unusable noise.
It really is. And importantly, the analysis also showed popEVE had limited to no bias across diverse ancestries in Nome rare variant scoring, which is a major win for equitable genetic testing.
So we have VDP telling us what system is affected, and popEVE telling us how badly it's affected compared to every other possible mutation. Let's talk about how this translates to, real-world diagnosis acceleration.
In a clinical setting, both models just significantly improve the interpretation of patient sequencing data. The prioritization power of V2P's phenotype-specific score, for instance, is undeniable. They tested it on 116 patients suffering from rare immune disorders. When they used V2P's score to rank the potential causal variants, the median causal variant ranking was dramatically reduced to 2.
2? That's incredible. What does that actually mean for a busy clinician?
It means finding the needle in a stack of 2 variants instead of digging through 10. Compare that to older established methods like CAD, where the median rank was 5.5, or CAPAS at 10.
That's a huge difference.
It is. Going from finding the causal variant in the top 10 down to the top 2. It drastically accelerates diagnosis, it reduces the need for expensive follow-up testing, and it potentially saves months of agonizing diagnostic uncertainty for families.
And popEVE offers a solution for what is arguably the biggest logistical and financial headache in rare disease diagnostics, the singleton case, where the child is sequenced but the parents are not available, or it's too expensive to sequence them as a trio. Wait, so popEVE can effectively replace sequencing the parents. That's a massive cost reduction for families and for hospitals. But how reliable is that ranking when you don't have the parental context?
It is extremely reliable, which is why this is such a powerful finding. When you don't have parental data, you lose that easy ability to filter out common inherited variants and zero in on the crucial de novo mutations, the new mutations that only exist in the child. popEVE solves this. It predicts severity so accurately that for diagnosed severe developmental disorder cases, it ranks the causal de novo missense mutation as the most deleterious variant 98% of the time.
98%.
Yes, it effectively ranks those critical severity-driving de novo mutations above all the rare inherited variants in the same patient. It just proves its utility even when parental data is missing.
And that capability isn't just for diagnosis. It must be a powerful discovery tool as well. What did the researchers find when they unleashed this severity metric on these large cohorts?
Its predictive power led them directly to the discovery of 123 novel candidate genes for severe developmental disorders, or SDD, in a large meta-cohort. Then the beauty here is they didn't just find these candidates, they could structurally confirm their importance. They found that 91% of the deleterious variants in these new candidate genes were physically close. We're talking within 8 angstroms of an interacting biomolecule in the 3D protein structure.
So that 8 angstrom proximity, that's the smoking gun, isn't it?
It is. It means the mutation is physically located right next to the critical region where the protein interacts with its partner, whether that's another protein, a signaling ligand, or nucleic acid.
So it's blocking a key interaction.
Exactly. This structurally confirms the mutation is not just some random noise. It's physically blocking or disrupting a key molecular interaction that's essential for function. And on top of that, these novel genes are functionally very similar to known DD genes. They showed strong enrichment for essential activities like chromatin organization and nervous system development, and notably, they had significantly higher expression in the developing fetal brain, which further supports their role.
That is just a phenomenal combination of computational power and real biological validation. So, to summarize the core achievement here, VDP fundamentally improves interpretation by introducing phenotype specificity, helping us define what a variant might cause. And then popEVE introduces this proteome-wide severity calibration, clarifying how bad that variant might be compared to others across every single gene. Together, they represent a huge leap forward in our ability to solve complex, ultra-rare genetic disorders.
They absolutely do, but we should probably note the limitations that they acknowledge because they do point toward the next frontiers of research. For V2P, its current reliance on those 23 broad HPO categories is mainly due to sample size. Extending this to predict more granular, more specific phenotypes, that remains a big computational challenge. And for popEVE, we mentioned this earlier, but it is critical. It currently cannot evaluate or compare the severity of a missense variant directly against a major loss-of-function mutation, like a nonsense or a truncating mutation.
Right.
And that remains a key barrier to achieving a truly unified, single-scoring system for all genetic risks.
That structural comparison is still the frontier.
It is. And finally, the developers of popEVE raised a really crucial, broader point about the sustainability of machine learning. They emphasized the high computational and, frankly, environmental cost of constantly creating new, monolithic, large models. popEVE's modular approach, repurposing existing models and adding a human-specific layer, it really demonstrates the value of using efficient, reusable components over constantly starting from scratch.
It's called for efficiency in genetic discovery.
Exactly.
That's a powerful note on sustainability and efficiency. So here's a final thought for you to chew on. Given that V2P has already pre-computed scores for every possible single nucleotide variant in a cell, mapping them to 23 broad disease categories, and popEVE has provided this high-resolution proteome-wide severity score, we are clearly moving toward a universal variant score. But, as the developers noted, achieving this goal responsibly means we need to rethink our current machine learning architecture. So how do we ensure that the next generation of predictive genetic models isn't just powerful enough to estimate a person's lifetime risk profile across all major genetic diseases, but is also computationally sustainable and accessible globally, so it can guide precision preventative medicine right from birth? Think about it. We'll catch you next time for the next deep dive.